Okay, hello and welcome to our webinar hosted by Lambert iGEM. I am Manasvi and
Sorry, it's muted. And I'm Neha.
we're going to be talking about the restriction enzyme cloning webinar. Thank you for coming out today. We greatly appreciate taking your time out to watch our webinar. And there's going to be certain things we talk about in this slides that won't make sense at first, but as we get through the workflow, it will make more sense. So just hold on with us.
And if you do have any questions throughout the session, please directly message Manasvi. She's the other presenter, and we will be glad to answer questions throughout the series. Thank you all for joining us. Let's talk about some of the objectives of this session today. So one, we want to understand how to build an effective plasmid and insert. Two, we want to be able to explain each step of the cloning workflow. And three, we want to know how to apply this knowledge in iGEM projects. So I'm sure you all are aware that iGEM is a synthetic biology competition. So the skills that we teach you today will give you a great head start in creating your own project. All right, so let's start off with a plasmid structure and what a plasmid is. So this is the sequence of DNA that your team is designing for your competition. And on the left right here is a quick diagram of a plasmid that I created. So we're just going to talk about each of the parts. So let's start with the origin of replication. This is that blue part right here. And this is a sequence of DNA at which replication is initiated. So this is where helicase, the enzyme, will go and unzip your DNA sequence to essentially separate your double strand and create two single-stranded pieces of DNA. And that's where your other enzymes, such as DNA polymerase, will be able to essentially duplicate your DNA. So some strands of DNA do have multiple origins of replications. So I want you guys to take a second and type in the chat why you might think that is.
Could you please repeat your question?
Yes. So some strands of DNA do have multiple origins of replication. So why do you guys think that is? And you guys can type in the chat what your guesses are. All right, so we are getting some mixed responses about this. Some people are saying maybe to make the plasmid more compatible with hosts. We also have time constraints in replication. So I want to go over this real quick. So sometimes your DNA strand is really long. So it takes a long time for one single helicase enzyme to start at one single origin and unzip all of this DNA. So if you have multiple origins of replication scattered throughout, it basically expedites the process and makes duplicating DNA much faster. So great job on that question. Next, we're going to talk about the antibiotic resistance gene. Sometimes this is called a selectable marker, and this is the gene that allows for selection of plasmid containing bacteria. So when you're essentially making your insert, which is the gene that you are designing, I'll get into that in a second, you're going to want to attach an antibiotic resistance gene just to weed out any of the bacteria that don't have your insert. So it's important that you have this just as a selection protocol. And sometimes you will see multiple, like in this plasmid right here, we have one up here and one down here. And sometimes you will have more than one inserts. So just based on the number of inserts you will have that according number of antibiotic resistance genes. Next, you will have your promoter, which is this purple section right here, and that's the site where the transcription of the gene is initiated. So right now we are in the DNA stage. At the promoter, your RNA polymerase enzyme will bind to it, and that's where transcription will start. So that's where your DNA will start getting converted into RNA. So we have this concept called promoter strength, and essentially the strength of your promoter determines or it regulates the rate of transcription. So the greater the promoter strength, essentially, the faster that your DNA will get converted into that RNA. And so in iGEM, typically we use a promoter with a high strength, which essentially means that you just get a larger amount of RNA produced. So these three things that I just talked about right here make up the backbone portion of the plasmid. We'll get into that in a second, which is just this gray portion right here. So again, that's what we call the backbone of the plasmid. And now let's talk about the other portions of the plasmid. So I'm going to skip restriction sites for now, and we're going to go down to the bottom of this like orangish color. This is called your insert or your target gene. And this is the sequence of DNA that your iGEM team is designing when you make your project. This is the sequence of DNA that your iGEM team is designing, and you're trying to get it into this backbone to create this giant plasmid. So to do that, you use these things called restriction sites, which again, we will get into more detail later. And this is essentially that intermediate buffer zone where you're able to cut up different fragments of DNA and glue them together to create your own custom fragment of DNA. So I see we have a couple things in the chat. I'm going to read them off. Okay, yes, so people are just answering the question I asked earlier. Any questions on plasmid structure before I move on? I'll give you guys a second to ask if you have any. Okay. So now let's talk about reading the backbone portion. So again, the backbone is this gray area that I was talking about earlier, and backbones are typically named PSB, a number, a letter, and another number. So let's talk about this PSB portion first. This just refers to the plasmid family. In iGEM, all the plasmids that we use will start with PSB, but if you run across another plasmid that was not developed by an iGEM group, it may say something else, but since we all are in iGEM, we're just going to make the assumption that all your plasmids will start with PSB. Next, you'll have two numbers. You'll have one on either side of the letter, and this shows your copy number. So this is how fast your plasmid will be duplicated. So if you see 1x3, that means there's a high copy number, which just means there will be a lot of plasmid produced. If you see 3x5, that just means the medium copy. And 6x1 means a low copy. So that just means like not that much of your plasmid is produced. Typically in our lab, we like to use a promoter with a great promoter strength and a high copy plasmid, so a 1x3 plasmid, just to make sure that we're getting as much yield as possible. But there are certain instances where we will use like a 3x5 or 6x1, but in our lab, those are not as common. And so now let's talk about this letter right here. This signifies the antibiotic. We'll again talk more about the antibiotic resistance later, but this is how we're going to weed out our bacteria. And the only ones that are going to survive are the ones with this antibiotic resistance. So in iGEM, there are 4 antibiotics. There's C, which stands for chloramphenicol, K, which stands for kanamycin, A stands for ampicillin, and T stands for tetracycline. So again, these are the 4 iGEM antibiotics. If you ever encounter an iGEM one, you will only see those 4. So for example, I'm going to go through a quick scenario. If you see PSB 1C3, well, you know that since it says 1C3, it's a high copy plasmid. Since the antibiotic is C, you know it's chloramphenicol. So this plasmid backbone is a high copy chloramphenicol backbone. That's kind of how you read these plasmid backbones. So real quick, we have a question for you. So which of the following plasmid structures allow for gene selection? A, promoter, B, restriction site, C, antibiotic resistance gene, or D, the insert gene? And feel free to type in the chat what your guess is. And we will go over the answer in about a minute. Okay, so most of you are getting this right. Yes, it is C, the antibiotic resistance gene. Real quick, I want to go over the other options. So the promoter is the site where transcription is initiated. The restriction sites are kind of that buffer zone that allow you to glue together different strands of DNA. Your antibiotic resistance gene is again the one that allows for gene selection. And your insert gene is the sequence of DNA that you as a team are designing. All right, so now let's talk about how to choose the right antibiotic. So again, this antibiotic resistance gene goes in that backbone. So the PSB1C3, for instance, we talked about. And the purpose is that antibiotic is used as selection. So only cells that absorb that plasmid that you are designing will have that antibiotic resistance. So only those cells are going to survive in the presence of that antibiotic. So you're going to want to choose an antibiotic for your plasma that's not already naturally present in the cell that you are transforming into. So I'll give you guys a couple of examples so that you guys understand what I'm trying to say. So for instance, DH5 alpha E. coli cells are a type of E. coli cells that we use in our lab a lot, and they typically don't have any antibiotic resistance. So pretty much in the presence of any antibiotic, those cells will not survive. So in your plasmid, you can use any antibiotic you want to. And again, if you put, so let's say we have that 1C3 backbone, which is chloramphenicol antibiotic. Let's say that my plate has chloramphenicol antibiotic, so only the cells with my plasmid will survive, and the cells that are not naturally resistant will not survive. So if I go into another example, let's say my cells are naturally resistant to tetracycline. I'm not going to choose a tetracycline antibiotic since all of them will survive. So I'm going to choose some other antibiotic. Let's say I choose chloramphenicol again. So if I grow these cells in the presence of chloramphenicol antibiotic, the cells with my plasmid that are resistant to chloramphenicol will survive. But my natural E. coli cells have tetracycline resistance, so they will not survive in the presence of chloramphenicol. I know this is a little confusing, so if you do have any questions, please feel free to type them in the chat below. And here in the bottom is a little tip that we would like to share from our team. So typically factories and other producers use ampicillin as their antibiotic. So over time, evolutionary adaptations have occurred and bacteria has become naturally resistant to ampicillin. So there is another antibiotic that we use called carbonicillin. It's interchangeable with ampicillin. It's a lot more expensive, so we don't use it in our lab as much. But essentially, when you're growing cells on a plate, there will be contamination. There will be mold spores, there will be fungus, they might be other types of bacteria. So you're just trying to get rid of all of this contamination. So you might want to put down another antibiotic such as carbonicillin right here on your plate, just so that it kills off any of the things that you don't want, such as mold spores, anything that just impedes with your cells growing. So it's kind of how you want to choose the right antibiotic. And we have another question. So we have not talked about this yet. We'll talk about this on the next slide, but I want you guys to take a guess. So which of the following initiate transcription? A, your promoter, B, ribosomal binding site, C, the start codon, or D, the terminator. So you guys can type in the chat what your guesses are again, and I'll go over them in a minute. Okay. We are getting some mixed responses for this one, so I'm going to go over it in the next slide. The answer is A, promoter, but I'll go over why in a second. So here's the insert. This is the portion of DNA that your team is designing. So here are the parts that make up an insert. You have your promoter, ribosomal binding site, start codon, gene of interest, stop codon, and terminator. So your promoter and terminator go hand in hand. These two regulate transcription. Your promoter is where transcription starts. So when we start off with the DNA sequence, that's where RNA polymerase will bind to, and it will convert all of your DNA into RNA up until it reaches the terminator right here. And after the terminator, RNA polymerase will fall off. And essentially the sequence that's in between these two are what's going to be converted into RNA. Next, you have your ribosomal binding site. So when you're going from protein expression, you have to go through transcription and translation. So now we've completed the transcription step and now we're on to translation, and your ribosome is the organelle that's going to assist with translation. So that's essentially converting your RNA into protein. So your ribosomal binding site is where essentially your ribosome will bind to this strand of RNA. And down here, your start codon and your stop codon are interchangeable again. Your start codon is where your protein expression will start. So the ribosome will start translating your RNA into protein, and it will translate all of this, and then your ribosome will fall off at the stop codon. And this portion right here is that protein that you're interested in. So when you're choosing your gene, typically you want to choose a fluorescence gene. This is so that you're able to distinguish your colonies once you start growing them. So in our lab, we usually use green fluorescent protein. It's just a protein that fluoresces green when this process occurs. So when your ribosome translates this into protein, it will just produce a green pigment. And it's kind of how we differentiate our cells that have this insert versus our cells that do not. So now I just want to talk about a quick overview about the exact steps of the restriction enzyme cloning workflow. So there are 7 main steps, and we will get into this in more detail. So you have to start off with hydration, then digestion, ligation, transformation, mini prep, nanodrop, and sequencing. And again, we will go over all of these in much more detail. So in the bottom is kind of what we're trying to do here. You will have a backbone that already comes with an insert. So every year, iGEM headquarters typically sends out a well kit. It's just a well plate with a bunch of different type of plasmids that you can use. And our team typically uses the RFP plasmid. This stands for red fluorescence protein. And it has a backbone with that red fluorescence protein insert gene that we talked about. And you will also have your gene of interest that you will order from a DNA company. When you're designing your DNA, there are several softwares that you can use. iGEM typically uses SnapGene, and that's just a software where you can plug in your ACs, Ts, Gs, make your own DNA sequence, and then when you want to order your sequence, you'll send that to a company. It's also affiliated with iGEM, and they will mail you your gene, your DNA sequence. And so what we're trying to do in this cloning workflow is get rid of that RFP insert and put in our gene of interest to create our target plasmid right here. And then eventually after we have this plasmid, we are going to put that inside of our competent cells. So now let's talk about more of the details about each of these steps.
Okay, so now we're getting into the actual workflow, the actual cloning workflow. So once you get your DNA, they are going to be dehydrated, as Neha said. So the first step is going to be hydration. And instructions for this are going to come with the DNA that you ordered. And it's a fairly simple process. You're just going to be adding water to your dehydrated DNA. Once you've done this, you're going to be going into restriction digest. And this can be split up into 2 different steps: your digestion and your ligation. So in digestion, you're going to be using restriction enzymes, and these are DNA cutting enzymes that basically just cut the plasmid or your DNA sequence into 2 separate sequences. And you can see on this plasmid right here, which we have taken off of SnapGene, it's a 1C3 backbone, meaning that it is a high copy chloramphenicol resistant backbone. If you see all of these like words all around, these are all different types of restriction enzymes. And based on where they're located is where they're going to cut. So looking at PST1, we know that it's going to cut right about there, right before the biobrick suffix. And just taking any of these is where the line meets up is going to be where it's going to cut. And which one you're going to use is going to depend on where you want to insert your target gene. Or if you were trying to separate the gene from the backbone, then you would have a restriction enzyme that cuts at the intersection of the 2. Restriction enzymes can produce either blunt ends or sticky ends, which we'll talk about more in the next slide. And after you do your digestion, you're going to want to run a gel electrophoresis, which we'll also talk in more detail about later. And this is to help you check your base pair length. This is to check your base pair length. So if you if you were doing a restriction digest and we're expecting to get 1400 base pairs, then on your gel electrophoresis, it should read 1400 base pairs. If it's like 300, then you know that something has gone wrong in your digestion. Okay, so this is a more in-depth of sticky ends versus blunt ends. So on the left side here, we have sticky ends, and these are going to be cohesive and have single-stranded overhangs. So basically, when restriction enzymes cut the sequence, they're going to produce almost like a stair. And this is so it has overhangs on both of the separate DNA sequences. So any sequences that have complementary overhangs are going to be able to form temporary hydrogen bonds between the nucleotides. So then they can bind together for a short amount of time. On the other side, we have blunt ends, and these are non-cohesive. When restriction enzymes cut these, it's more in a straight line. So they can't actually bind to each other. They just kind of have to come together by chance. Okay, now this is the second step of the restriction digest, and this is ligation. In ligation, you're going to have DNA ligase, which is an enzyme, and it's going to covalently join the phosphate backbone of your DNA. And it does this by forming a phosphodiester bond. So before, how I was saying that they're going to form a temporary hydrogen bond, a phosphodiester is more permanent, and it will make sure that the plasmid and the backbone and the insert are going to stay together. And for ligation to occur, you need to have your sequences to stay together long enough for the ligase to join them. And this is why sticky ends can be much more easier to work with than blunt ends, as sticky ends are more likely to stay together as opposed to blunt ends, which it is less likely for them to stay together long enough for DNA ligase to join them. Okay. So when you do your restriction digest, there are multiple outcomes that can happen. So let's make a scenario where we have 3 different DNA sequences. We have our backbone, we have our original gene, so this is the gene that came with our backbone, and then we have our desired gene, and that's what we're trying to insert into our backbone. Now, when we do our restriction digest, there are 3 possible outcomes. So we can have the backbone ligating to itself, the backbone ligating to the original gene, or the backbone ligating to the desired gene. And these can have visual differences on your petri dish. And a petri dish is just like a plate in which your cells can grow on. And what you're seeing on the right there is what is going to be seen after you do your transformation, which we're going to talk about in more depth in the next few slides. The amount of outcomes you have can also vary depending on the amount of inserts you have. So the more inserts you have, the more outcomes or possibilities you have. And the type of insert gene also makes a difference. So if I were to use, if both of my genes had RFP in them, then both of them would show a red fluorescence on my plate. So it just depends on which selection genes I use and which, blue-white screening, fluorescence, proteins, anything like that, will have different visual effects on my petri dish. Okay, and we have another quick question for everyone. How does restriction digest differ from ligation? So A is restriction digest needs DNA as opposed to ligation, which needs competent cells. B is that restriction digest cuts sequences, whereas ligation joins sequences together. C is that restriction digest deals with sticky ends, whereas ligation deals with blunt ends. And D is that restriction digest checks base pair lengths, while ligation degrades DNA. So just put your answers in the chat and we'll just give everyone a minute. Okay, good. For the most part, I'm seeing everyone put B, but there are a few mixed ideas. So the correct answer was B, restriction digest does cut the sequences while ligation joins them together. A would be wrong because both of these sequences are only dealing with DNA. We haven't even gone to the actual use of competent cells. This is used later in transformation. And C is wrong because restriction digest and ligation, they both deal with sticky ends and blunt ends. This is purely based on your restriction enzymes, and you can use both for both of them. And restriction digest does not check base pair lengths, it separates it, and ligation does not degrade your DNA.
All right, so now let's talk about transformation. So this is the step where you have your desired plasmid that we talked about. Let me go back a couple slides. So this plasmid right here, where you have your backbone ligated to your desired gene, once you have that, you are trying to transform these into competent cells. So competent cells is basically a fancy way of just saying your bacteria. You can use E. coli. Some other labs do use different types of bacteria depending on what type of experiment they are running. So the three main steps of transformation are to add your competent cells, also known as your bacteria, and your plasmid, so that's the DNA sequence that you designed, into a transformation tube and let that incubate for about half an hour on ice. Next, you're going to want to shock your cells to basically allow them to open up their pores in their membrane so that DNA can be absorbed into your cells. And then after you do this, you're going to want to place your transformation tubes in the shaking incubator. So last, you're going to want to plate the cells onto antibiotic plates to let the cells grow for 24 to 48 hours. And this antibiotic that you put on the plate is going to be that same antibiotic in your backbone. So if your backbone had chloramphenicol resistance, you're going to want to make chloramphenicol agar plates to grow your cells on. And after your cells grow, they might look something like this. For example, here's the plate, and you might see different colors of colonies. So kind of as a gauge before we move on, this is kind of what you would see. So if you see clear-ish colonies, that can actually, let me backtrack a second. So it really depends on what type of plasmid that you initially start off with. So in our lab, we start off with plasmids that have an RFP or red fluorescence insert. So we talked about how there might be three outcomes of ligation. If you do end up having your backbone ligating to its original gene, such as RFP, you are going to see red on your plate. If you have your backbone ligated to your desired gene, in our lab, we typically use green fluorescence protein. You're going to see a greenish tint. Now, it's important to note that sometimes your cells haven't been growing long enough for fluorescence to occur. So a lot of times you may just see clear whitish colonies, and that's okay. You're just going to want to pick up those white colonies. And if you see any hint of green in your colonies, you're going to want to pick up those two for our next steps of transformation. And when you're preparing your competent cells, we're going to go over two main ways to prepare competent cells, but just make sure to follow the protocols that are supplied by the company that provide you with those competent cells. So now there are two main types of transformations that people do in their lab. There are chemical transformation and electroporation. So I'm going to talk over chemical transformation first, and then I'll move on to electroporation. So in chemical, when you're preparing your competent cells, you're suspending your bacterial cells in a salt solution to create a cation bridge. Let me break this down for you. So your membrane of your cell is made up of a phospholipid bilayer. Those phosphate groups in that bilayer make it very negatively charged. DNA also has phosphate groups in its backbone, so DNA is also negatively charged, and like charges repel. So it's very challenging to get DNA to enter your enter your cell traditionally. So when you suspend these into a salt solution, we typically use calcium chloride salt. When we're preparing chemical competent cells, the salts will dissociate and create ions. So the positive calcium cations will bind with the membrane and essentially neutralize the membrane so that it's much easier for your negatively charged DNA to enter the cell. So after you have your competent cells prepared, you're going to move on to the heat shock step. So the membrane is selectively permeable, which means that it only allows certain things to enter and exit the cell. So the purpose of the step is to increase membrane permeability, just to make it more fluid and open up those pores wide enough so that the DNA plasmid can enter through. So we heat shock at 42 degrees Celsius. We did incubate on ice, so they are very cold. And now that we're bringing it up to 42 degrees Celsius, this is a rapid heat shock that essentially rapidly increases membrane permeability and opens your pores for a couple of seconds for your plasmids to slip through. So some pros of chemical transformation is that the results are fairly predictable and your DNA concentration can be increased or decreased as needed. So if you notice that your cells aren't absorbing that many of your plasmids when you grow them on plates, you are able to adjust the DNA concentration and add more if you need to. One of the biggest cons of chemical transformation is that it takes a really long time and that it has a low transformation efficiency. So this means that just not that many of your cells are absorbing up your plasmid, and you're going to have to redo this multiple times. So now let's talk about electroporation. Your competent cell preparation is pretty much the exact opposite of chemical cell preparation. So in electroporation, you're not heat shocking. You're running an electric current through the cells to open up the pores. and salts do conduct electricity when they are in their ion form. So when we're preparing competent cells, we want to wash all the cells in a nonpolar solution called glycerol to essentially get rid of all medium salts, and this reduces the chance that your cells will conduct electricity because you really don't want to electrocute your cells. So now we're going to talk about the electroporator, which is this machine that you put your cells into and essentially runs an electric current through the cells to again increase the voltage across the membrane, which again increases the membrane permeability and opens pores to allow DNA to pass through. And for electroporation, we typically perform that at freezing temperature at 0 degrees Celsius. This is just so that we don't want to deal with any additional heat interfering and possibly damaging the cells again. So some pros of electroporation is that it is less cumbersome and relatively easier than chemical transformation, and you do have higher transformation efficiencies. So there is a higher chance that your cells are able to absorb your plasmid. But one of your big cons of this is that it is way more expensive. Purchasing electroporated competent cells are very expensive. You also need specific machinery to do the electroporation part itself. And again, this is extremely sensitive to salt. So you can't adjust the DNA concentration like you could in chemical transformation. So we typically use smaller portions of DNA when we do electroporation, just again, so that we don't end up accidentally electrocuting our cells.
Okay, now we're going to be talking about colony PCR, and this is just going to amplify your DNA or your cells. And before you can run a colony PCR, you have to dilute your colonies because colonies are made up of a bunch of cells. And in order for your colony PCR to function properly, you're going to want a low and even concentration of your cells. Your reagents for colony PCR are water, DNA, forward primer, reverse primer, and master mix. And master mix, for any of you who don't know, is just a combination of all the other small reagents that you need for colony PCR, and it just makes it less tedious and an easier process. And your forward primer and reverse primer are just going to bind to your DNA, and forward primer reads forward, reverse primer reads backwards. And whatever they read between the two primers is what is going to be amplified in your colony PCR. So now let's talk about the actual colony PCR process. So there's three main steps to it. There's denaturation, annealing, and elongation or extension. In denaturation, the double-stranded DNA is going to separate into single strands. In annealing, your primers, which is your forward primer, reverse primer, are going to bind to your selected regions, and you can choose where they're going to bind. And then in elongation, a DNA polymerase is going to bind to the primers and add nucleotides, essentially duplicating the DNA strand. And this process will happen a lot of times and creating an exponential growth for the DNA. And you're going to want to use a thermocycler for your colony PCR just so it can cycle through different temperatures, and this makes sure that all the processes are occurring. Also, when you are adding your reagents, and this is kind of like you can apply this to all protocols, but you're going to want to add it from least expensive to most expensive. So in this case, you would want to go from like water, which is least expensive. And this just makes sure that if you do mess up, you're going to be saving the most money. And here is a quick poll question. What is the purpose of transformation? A, DNA amplification, B, create competent cells, C, gene selection, or D, add plasmids to your competent cells. You can just put the answers in the chat and we will go over the answers real quick. Okay, yes, good job, everyone. The answer is D. It is adding plasmids to your competent cells. Just like we went over in transformation, you're going to be opening up the competent cells so that it can take up the plasmids. Yeah, and then we have another question. It is a double poll. This question is, how is colony PCR verified? So we have not talked about this yet, but we will in the next few slides. So just guess. So there's 4 answers: transformation, inoculation, gel electrophoresis, or does it need to be verified? Just put your answers in the chat.
again, we have not talked about this yet, so don't be afraid to get this question wrong. Just make your best educated guess.
I actually have a question. I didn't understand the difference between colony PCR and just normal PCR. In general PCR. So what is role of cell being there, like colonies of cell being?
So we're just going to fact check that real quick. But regular PCR is just to amplify, just normal strands of DNA. Colony PCR is just to verify that your transformed sequence of DNA is amplified. And typically in colony PCR, you are amplifying physical cells with your DNA inside, rather than just your DNA strand alone. But we will fact check that and get back to you on that question.
Okay, so most of you, sorry, one second, let me just pull up the chat. Yeah, okay, so most of you did put C, and that is the correct answer. Gel electrophoresis is how the colony PCR is verified, and we're going to be talking about that in the next slide.
Okay, so now we're going to go into the details of gel electrophoresis. So the purpose of this is to separate DNA fragments according to size, and this is used to check the success of your colony PCR. So there are multiple reagents that are used in this. You have a buffer solution. So this basically allows your gel to evenly conduct electricity, because again, this is a chamber that does conduct electricity. So you do want to make sure that you are conducting it evenly. Someone said voice break. Are you guys able to hear me clearly? Could you please just type yes in the chat if you can hear me? Okay. Okay, is this better or is it still breaking up? Better? Okay. Thank you for
Also, you will have to repeat the difference
yes.
between colony PCR and, because I missed you there. There was
Okay.
voice cut.
Okay, sounds good. So essentially the difference between the two, Manasvi, please interrupt if I am incorrect, but the difference between the two is that PCR, just regular PCR, amplifies strands of DNA alone. And in colony PCRs, we are amplifying cells that already have DNA in them. So in both of them, we are amplifying DNA sequences. Just in traditional PCR, it's just the DNA alone, whereas in colony PCR, the DNA is already inside of a cell. So was that helpful?
Yeah, thank you. Thank you.
Of course. And yes, guys, feel free to interrupt me if you guys have any questions. So I'm going to restart with the gel, just in case anyone didn't catch me the first time. So the purpose of a gel is to separate DNA fragments according to their size, and it's essentially used to check the success of your colony PCR. And you will have multiple reagents that you will use when you are performing a gel. So you have a buffer solution. So when you're doing gels, you're running an electric current through this device that we'll talk about in a second. And your buffer just makes sure that your gel is evenly conducting electricity. And so again, which buffer you use typically depends on your cells and your DNA fragments that you're using. So it will change. The buffer that we use in our lab is TAE buffer, and we typically use it as a 1 molar concentration. So that's what this X means. It just means 1 molar concentration. But again, your buffer solution will differ based on the type of DNA that you are using. Next is agarose. So this is the substance that actually makes up the gel, and it's a very hard substance that forms. Actually, it's not that hard. It's kind of like a gel-like substance, and it just forms a lot of pores that DNA can travel through. Next is your DNA stain, and it's a highly sensitive stain that's used for the visualization of DNA. So DNA naturally is very clear, so you're not able to see it. So when you mix it with the stain, you're basically just able to see the bands of DNA on a gel. And I'll show you guys an example of that in a second. And finally, you have your ladder, which is used to approximate the size of your DNA. Your ladder is just a reference point. And since each gel is different because agarose is different, no two gels are going to be exactly the same, it's really important to have your ladder. Otherwise, you're not sure what band you're looking at without a ladder. Oh, okay, maybe my internet broke out for a second. Okay, let's try that again. Okay. So we have a gel chamber. So this is kind of the chamber that you put your gel inside of. So this blue part right here is the gel itself. This is the agarose portion. And these dark blue wells in the corner are your wells where you are going to pipette your DNA into. So it's really important, I can't emphasize this enough, in which direction you put your gel in the gel box. You always want to make sure these wells right here are in the side with the negative electrode. This is due to like charges. So again, DNA is negatively charged. So you want to start it off at the negatively charged end since like charges repel. You want the bands to flow this way. So if you're putting them together, your like charges are going to repel and your DNA is going to want to move to this direction. Whoops, sorry. And there's this positive electrode right here. So your DNA is also attracted to the positive electrode. So you just want to make sure that in our lab, we use this saying called runs to red. So whenever you set up your gel, you want to make sure that your DNA well is running towards the red positive electrode. And if you accidentally flip your gel, so let's pretend that this part right here was the positive electrode, your DNA is going to be attracted to that and it's going to basically run off the gel and you're not going to see any bands. So this is why it's really critical that you put your gel in the right orientation. This box right here, does someone have a question? Sorry. Okay. So this box right here is your gel chamber itself, and this is a hollow box that has this is where you pour your buffer solution that we talked about, and it essentially like covers the gel with buffer. So just to allow that electricity to flow evenly. And when you're running a gel, there will be like bubbles popping up on the side. It will be like large like streams of bubbles. It will just be like a vertical stream. And if you see these bubbles on the side of your gel, it will be on both sides. That means your gel is running correctly. If you don't see those bubbles, I would double check with your power supply and make sure that it is plugged in properly. So this power supply is what controls your voltage, your amperage, and your time. So your voltage is essentially the pressure that the electrons are flowing through your gel chamber. Your amperage controls the volume of electrons that are flowing through, and your time obviously controls the time that your gel is on and running. So to connect the gel chamber to the power supply, the company will provide you with wires. You'll want to match up the color. So the black wire connects with the negative electrode and the black port in the power supply. Red wire connects to the positive electrode and red port. And again, you will have different variables that affect the way your gels run. So your voltage and your amperage will change per gel. Again, this really does depend on who you buy your gel box from. They will have different values that you will use. And also real quick, so your gel, we talked that it's made of agarose. So you can have gels that are different percentages of agarose. Typically in our lab, we use 1% agarose in our gel, but based on the size of your DNA fragments, you will need gels of different thicknesses. So if you have a gel if you are trying to run a gel on DNA fragments that are very large, larger fragments don't travel through as easily. So you're going to want to create more pores so that the fragments can be further separated by size. So if you have larger fragments, you're going to want to make a gel that has a small percentage of agarose. And vice versa. If you have small fragments, they can wiggle through those tight spaces easier. So in order to greatly distinguish the bands, you're going to want to make a gel with a higher percentage of agarose. So large fragments, less agarose, small fragments, more agarose. And this essentially just allows you to separate the fragments based on size. And again, this is why it's really important that you have that ladder right here. because based on the percentage of agarose, your ladder will look differently. Sometimes it will look like this. If you have a very thick gel with a large percentage of agarose, sometimes your ladder will be squished, and maybe like this whole portion will be your entire ladder, and your DNA fragments will be much closer together. Maybe you have a gel that is very small percentage of agarose, your DNA ladder will be longer, and you will want to use a bigger gel for that. So again, your ladder is your reference point, and the company you buy your ladder from will give you a reference. So they will tell you how many base pairs each band length is. So for instance, this band is about 3,000 base pairs. This band right here is about 1,500 base pairs. This band is about 500 base pairs. And you use this with your fragments to determine about how long it is. So 1, 2, 3, and 4 are essentially DNA fragments that we've pipetted ourselves into these wells. So let's look at fragment 1. You can see that it's very close to the well, so automatically you can assume that it is a fairly large fragment of DNA. So you can see here that this says that this line is approximately 3,000 base pairs. Now, since it didn't travel as far, we know that it's larger than 3,000 base pairs, so we can make the assumption that this is going to be a larger than 3,000 base pair fragment. Let's move on to well 2. This band traveled all the way here. So since it traveled really far, we're under the assumption that it is very small fragment of DNA. So here is our next reference point. Obviously in your ladder, you will have reference points for each single band. Just in this example, we have 3 right now. So this band is approximately 500 base pairs. And since band 2 traveled much further, you can go under the assumption that this band is much smaller than 500 base pairs. Well, 4, you can see that it lines up with that 500 base pair length. So you can make the assumption that this band is approximately 500 base pairs. So sometimes you will see 2 bands in your gel. This typically occurs after digestion. When you're cutting up your plasmid into multiple strands, you might see multiple fragments of DNA. And this just makes sure that like you're able to know that you're digesting correctly. So this band is approximately 1,500 base pairs. This band up top didn't travel as far, so we know that it is bigger than 1,500, but it's not as close to 3,000. So we know that this band right here is between 1,500 and 3,000 base pairs. So now here are some examples of gels that we've run in our lab. So in the middle again is our ladder. This is something like what it will look like when your company gives you the ladder. You can see that each band has a distinct kilobase pair length. So this band right here is very vibrant. That's approximately 3 kilobases, so approximately 3,000 base pairs long. This band all the way down here is 0.5 kilobases. So like each of these bands will have different lengths. And here is our ladder again. You can see this band right here in the C4 well. So this band right here is going to be our reference point. This band is approximately 1 kilobase pair. And so we know that this band traveled slightly further than this band. So we know that this band is slightly smaller than 1 kilobase. If we want to choose a band up here, this is again our 1 kilobase reference. This band didn't travel as far, so we know that this band is greater than 1 kilobase. And sometimes you will see this smearing that occurs. Whenever you see smearing, it won't be one definitive line. It will be like kind of the smear that you're seeing here. And when this occurs, this typically means that your voltage is too high. So if this happens, you're going to want to make another gel, reload your gel with the same reagents, and just run the gel again at a lower voltage, and you should see more bands that look like similar to this. All right, let's move on to inoculation.
Okay, so if your gel ran successfully, meaning that the gel lined up with your ladder at the base pairs you expected to see, then you're going to move on into an inoculation. And this is to allow your cells to grow, and it's a fairly simple process. You only need 3 reagents: growth medium, antibiotic, and cells. So your growth medium is a medium for your cells to grow, and generally we use LB, but this can differ depending on your experimentation, whatever you're trying to characterize. Then you're going to have your antibiotic, and it's going to correspond to your backbone. So going on the 1C3 backbone, which is chloramphenicol resistant, we would be adding our chloramphenicol antibiotic. And this would just remove any of the cells that didn't take up the plasmid. Then lastly, we're going to be adding our cells, and this is going to be cells taken from our transformation, not from the colony PCR or the gel, because those are just checkpoints. We're going to be going back to our plate from the transformation, and we're going to be taking just a scrape of cells. And so if you were like using a pipette, you would grab just enough that you could see it on your tip, but it's a very, very small amount. And you're just going to add that to your solution. When you are doing a liquid culture, your growth medium to antibiotic ratio is 1 milliliter to 1 microliter. So if you're adding 5 microliters or milliliters of growth medium, you're going to be adding 5 microliters of your antibiotic. And you're just going to let this chill or rest overnight in an incubator. And the next day, you should see cloudy solution, and this signifies that your cells have grown.
Before we move on real quick, we do have a question. So why are we not performing a normal PCR, and what is the advantage of doing colony PCR here? So if we backtrack a couple of slides. So a colony PCR is a checkpoint after transformation. And in transformation, we've already put our plasmid inside of cells. So we're just taking that colony that grows. So let's say that this is our plate right here. We're just scraping one of these colonies and running it into a PCR tube. And so since this is already inside of a cell, we're just running a PCR on that, not on the DNA itself. Does that kind of answer your question?
Also, when you are doing a colony PCR as opposed to a normal PCR, you can choose a specific sequence. So using your forward and reverse primers, they can bind at specific sites on your plasmid. So you can like just amplify a certain sequence. So if you're trying to amplify your backbone, you could only amplify your backbone, or you could only amplify your insert. And generally, you're going to want to amplify your insert, and this will just make sure you have the correct insert in your thing. Because generally, your original insert that came with the plasmid and your desired insert, which you're trying to insert, are going to have two different base pair lengths. So by doing a colony PCR, you can see the base pair length. So if my desired base pair length is 1400 and I got like 1300, well then I know that probably I got the DNA plasmid probably like religated to the original DNA, if that makes sense.
So yeah, colony PCR is again just a checkpoint. We're kind of amplifying this DNA right here to run a gel on it, just to make sure that we have the correct sequence identified. And we are doing a colony PCR because it's colonies of cells. Typically, this is the only place where you will run a PCR on cells themselves rather than actual DNA strands. So I hope that kind of answered your question. If you do have more questions on colony PCR, we'll be glad to share resources on that. So let's move on.
Okay, so hopefully your inoculation went successfully, so you should be seeing cloudy cells or cloudy solution in the signifying that your cells have grown, or you might even see like a cell clump at the bottom. Then you're going to be running a mini prep on this. And this is just a series of wash cycles and centrifuging, which breaks down the cellular membrane and gets rid of all the extracellular debris. And what this is going to do is it's going to leave you with the purified plasmid DNA. Then you're going to use a nanodrop, which is a small machine in which you put 1 microliter of your DNA, and it will check the purity and concentration of your sample. An optimal range for your purity is 1.8 to 2, and an optimal concentration is above 100, but the higher the better. So the data you get from your nanodrop can be used in sequencing. And in sequencing, you're going to be sending out your DNA to a lab for them to sequence your DNA. And basically, they'll just tell you the ATCGs of your DNA sequence. So when you do this, you can align the sequence that you got from the lab to the original or intended sequence you have. So if you constructed your sequence on SnapGene, you can just compare it to that. And you can see if they both have the same sequences, then you know that everything went well. And if they have different sequences or significantly different sequences, then something probably went wrong and you want to backtrack. And so throughout this process, there are a lot of chances for mutations to happen or any problems or contamination that can cause your sequencing to not be accurate. When you are doing sequencing, you're going to want to just follow the company's instructions. So they'll tell you how much of your DNA and how much of any other reagents you should be adding, depending on your nanodrop data.
Real quick, so we do have a question, where can mutations occur? So when we are running things through the PCR machine or when we're amplifying DNA, sometimes nucleotides can be mismatched. So that's kind of where mutations can occur throughout this process.
Yeah, and then also like contamination, like we said in the beginning, can grow like bacteria, spores, or even like smaller things that you may not notice. Or even if when you are doing some of the protocols or going the procedures, you may mess up or not add the correct amount, and this could cause your process to not go as planned, or it would cause a mutation. Okay, so we have a quick question here. What does the nanodrop quantify? A, concentration and purity, B, base pair length and purity, C, purity and cell growth, or D, concentration and base pair length. So just put your answers in the chat and we will go over that in a minute. Okay, good job. Yeah, everyone has been putting A, and that is correct. And nanodrop will quantify your concentration and your purity. And remember, for concentration, you're going to want a higher concentration, preferably above 100. And for purity, you're going to want a range about 1.8 to 2. Okay, so you're still not done. You're almost done, though. We're in the final stretch. Once you've done all of that, you have done your sequencing and you know for sure that the sequencing lines up, you have to create a glycerol stock. So in order to create a glycerol stock, you have to do another transformation. So this is not the same as the one that you did originally after your restriction digest. This is using your mini prep DNA, so that purified plasmid DNA that you got from your mini prep. And you're going to be performing a transformation, allowing the cells to take up the plasmid. Then you're going to do an inoculation, allowing the cells to grow. And then you're going to create your actual glycerol stock. And to do this, you're going to be adding glycerol to your liquid culture cells. And doing this will allow your cells to be stored later and used for experimentation or any characterization you're going to be doing later in your project. And make sure to store your glycerol stock in a negative 80 degrees Celsius freezer. This is very, very important, because if you do not store it in that temperature, then it will degrade. And also keeping it in a negative 80 degrees Celsius freezer will promote stability of your cells. If you do not, then you won't be able to use them in experimentation, as they will be degraded. Sorry. Okay, so another question, where are glycerol stocks stored? I just went over this, so you should know the answer. There are 4 possible answers: refrigerator, negative 80 degrees Celsius freezer, room temperature, or just a regular freezer. So just put your answers in the chat. Yeah, good job. Okay, yes, it is negative 80 degrees Celsius freezer. And like I said, this is very important. You want to make sure that your glycerol stocks are in there at all times. And if you do need to take it out, make sure you keep it in an ice box or keep it as cold as possible to prevent any degradation of your cells. Okay, and one last thing. Throughout this process, a really great website is Adgene. They have protocols for everything we have talked about and more. They have helpful videos and step-by-step instructions. So when you are doing your own restriction enzyme cloning workflow, definitely check out this website. It is very, very helpful. Yeah, so definitely check that out. It's called Adgene. Okay, and we are almost done. So this is a QR code, if you could scan it, and it is very, very short. It's just for our pre-post survey. It has like a few questions just to see how much you've learned from this. And we're also going to go over some of the answers from that. So make sure to answer that and we'll go through that.
We will also put the link in the chat, and please stay on after you complete the post survey. We're going to go over some of the answers and a couple last tips and tricks.
Yeah. Also, if you do have any questions right now, would be the best time to put them in chat or to just unmute and say them. And I've put the link in the chat, so you can use that or you can scan the QR code.
So yep, we'll give you guys a couple minutes to answer, and after we see that the majority have answered, then we'll go ahead and go over the questions. Thank you to those who have completed the survey. We have about 4 responses, so we're going to hold off a couple more minutes before we go over them. While we wait for some more responses, would anyone be interested in another session on a different topic? Yes, okay, I'm seeing some yeses. What are some things that you guys struggle as in a lab? Because we noticed that the majority of you have only been an iGEM team for 1 or 2 years. So is there anything we can help with, maybe like understanding iGEM requirements on how to win a gold medal, or any like molecular biology topics? Or what are some things that you guys see yourself wanting more help with? Because we would love to help you in any way. And again, it doesn't just have to be wet lab. We can post them on like software, hardware, on human practices, just on any topic that you might want some more extra support on. So feel free to DM me or just type in the chat if you guys have any suggestions for future series.
Okay, so a lot of you have answered our post survey. Thank you for doing that. Now we're going to just go over the answers. So we do see a bit of mixed answers for the first one, which is the following steps is the correct order of the workflow. So the correct answer was A, and that is digestion, ligation, transformation, calling PCR, inoculation, mini prep, nanodrop, and sequencing. So just a quick overview of what this was, just a quick recap. First, you have to do digestion, which basically just splits up the DNA sequences. Then ligation will glue them back together. Transformation is when your competent cells will take up the plasmid DNA, and then colony PCR and gel are just like a checkup step or checkpoint step, sorry. And these just make sure that your base pair length is what it should be. So colony PCR just amplifies your DNA. Then you have inoculation, allowing your cells to grow. Then you use that for your mini prep, which gives you the purified plasmid DNA, and then a drop gives you data about your purity and concentration, and then sequencing will give you the actual DNA sequence of everything you have done so far. And then on the second question we asked was, what is the copy number and antibiotic for this plasmid? So we're seeing two answers for this one. Some of you are putting A and other of you putting C. So C was the correct answer. Good that everyone, oh, okay.
Sorry, I'm gonna go back to that slide.
Okay. So good that everyone knew that yes, T does stand for tetracycline. Good on that. But 3T5 means that it is a medium copy. 1x3 would be high copy and 6x1 would be low copy. So just keep that in mind. Okay, and that was everything for this webinar. We will be emailing everyone the recording, the screenshot, and the presentation. So be on the lookout. We'll do that either today or tomorrow. And thank you for taking the time for coming here and doing our post survey and doing all the polls. We greatly appreciate it.
And if you do have any more questions, feel free. Sorry if there's like noise outside. There are like lawnmowers outside running. But feel free to reach out to us at igem.lamb@gmail.com. And again, later today, early tomorrow, we will send out this presentation that we used today, as well as our recording. And we are getting a couple of questions. So we'll circle back with our advisor on some of your questions that you have, and we'll put any additional resources there as well. So yeah, thank you so much for joining. We really appreciate you guys staying here today. And when you're ready, you can leave the meeting. I'm going to stop the recording right now.